is Handsome ASMR, back with another video. This time we're going to dig into renal physiology, at least the overview of it. So let's get started. So renal function is another way to say kidney functions. And the kidneys indeed perform several important functions. These include filtration of blood in order to remove our waste products like urea and creatinine. It is also responsible for regulating blood volume and composition, including the composition of electrolytes like our sodium, potassium, and calcium levels. And then it is also responsible for maintaining acid-base balance, making sure that the number of H+ acidic ions and bicarbonate basic ions balance out, thereby regulating pH. It is also necessary for excreting metabolic waste products and foreign substances like drugs and toxins. And the kidneys, of course, have some endocrine functions, such as the secretion of hormones like erythropoietin, thereby regulating erythropoiesis, which is another way to say the production of red blood cells. It is also involved in secreting renin, which is a hormone needed for regulating blood pressure, and then calcitriol, which is a hormone needed for regulating calcium and phosphate balance. As such, kidneys are important for regulating blood pressure through the classic renin-angiotensin-aldosterone system, also known as the RAS system or the RAAS. They can also utilize pressure natriureases to complete this function. So now that we know about the kidney functions, let's review, or at least look over the kidney anatomy and microanatomy. So each kidney generally has an outer cortex and an inner medulla, where the cortex can be shown over here and then the medulla over here. And what we see over here is the functional unit of the kidney, which is called the nephron. So the nephron, as shown over here, consists of the renal corpuscle, which is composed of the glomerulus and the Bowman's capsule, as we see over here, and then the renal tubule, which is comprising of the proximal convoluted tubule that we can kind of see over here, the loop of Henle, which is shaped like a loop, also over here, and then the distal convoluted tubule, which would be located about here, and about here on this side. And then the collecting duct, which will be this large stem that we see traveling from the cortex to the medulla. So those are the parts of the nephron. The glomerulus is special because it is a capillary tuft that is responsible for filtering our plasma that flows to our kidneys. And so the glomerulus would thus be found in cells happen to be fenestrated in their structure, which is important for permitting passage of small molecules, but not large proteins. The next layer we have is called the basement membrane. This one provides a charge barrier, and it is important because it prevents negatively charged proteins from passing through. And then we have podocytes, which have foot processes that form filtration slits, which further limit protein passage. You can remember podocytes having foot processes by paying attention to the prefix podo, which is a root word for foot. The next principle is glomerular filtration rate, commonly known as GFR. So, pay attention here. GFR is the volume of plasma filtered through the glomerulus per the unit time. The typical filtration rate ranges from 120 to 125 milliliters per minute for a healthy adult. So the GFR, or glomerular filtration rate, is a key measure of kidney function, and it can thus be regulated by several factors. The first factor is the hydrostatic pressure. Such pressure is known to increase the filtration rate in the glomerulus. The next type of factor is the oncotic pressure. This happens to decrease the glomerular filtration rate. This is because the proteins in the plasma stay there instead of being filtered, and through the principles of osmolarity, the presence of proteins will draw more fluid or water towards it rather than away from it, which thus decreases the amount of fluid that is being filtered. In other words, more fluid tends to be kept inside the plasma rather than being filtered out into the renal tubules. And then we have afferent and efferent arterial tone. So, afferent arterial is the small blood vessel that goes to the glomerulus in order to be filtered, while the efferent arterial is the one that exits from the glomerulus. You can think about this as efferent having the same first letter as the word exit. Okay, so anyways, constriction of the afferent arterial decreases GFR, while constriction of the efferent arterial will increase the glomerular filtration rate. Now that we explored more about the glomerulus, let's now explore what happens after filtration in the glomerulus. So after filtration, the filtrate passes through the renal tubules, where reabsorption and secretion occur in sequential manner. The first tubule we have is the proximal convoluted tubule. This is where reabsorption happens. In fact, the proximal convoluted tubule is responsible for the bulk of solid reabsorption. Specifically, about 65 to 70% of filtered sodium, chloride, and water, and then ideally 100% of glucose and amino acids. There are several transport mechanisms involved in this reabsorption. First, we have the sodium glucose cotransport and the sodium amino acid cotransport. The sodium glucose cotransport is known as the SGLT transporter. And then, although it's not shown over here, you can expect the transporters to be present on this side. The next transporter, or in this case exchanger we have, is called the sodium hydrogen exchanger, also known as NHE. This is shown over here, and this is responsible for bicarbonate reabsorption. Because if you follow this step by step, pay attention to what happens when you have the sodium being reabsorbed and the H plus ion being secreted. This is paired with the action of secreting bicarb into the renal blood rather than to the lumen or the interstitium rather than the lumen. And so bicarb is generated for every H plus ion or proton that is being excreted. Okay, so besides that, we have active transport of bicarbonate through the action of carbonic anhydrase, shown over here, and then passive reabsorption of water through the aquaporins. So always remember that water will also get reabsorbed in the proximal convoluted tubuli. The next part of the renal tubule is the loop of Henle. So the loop of Henle generates a medullary osmotic gradient, which is essential for concentrating urine. It consists of a descending limb and an ascending limb. So the descending limb, it happens to be permeable to water, but not to solids. So water is reabsorbed, and the filtrate becomes more concentrated as it descends. On the other hand, the ascending limb becomes impermeable to water, but actively transports sodium, potassium, and chloride out of the filtrate, making the filtrate more dilute. The thick ascending limb happens to be the site for loop diuretics to do its function. And as we can see over here, we see the reabsorption of sodium, potassium, and chloride. And loop diuretics will target the specific channel proteins to prevent the reabsorption of these ions. And a classic example of such a loop diuretic is furosemide. Okay, now we have the distal convoluted tubuli, which comes after the thick ascending limb. So right about here, this is where the distal convoluted tubuli would be present. So the distal convoluted tubuli is responsible for fine-tuning sodium and potassium balance. So once again, sodium reabsorption happens here. Specifically, the distal convoluted tubuli reabsorbs sodium through the sodium chloride cotransporter and is thus regulated by aldosterone, which happens to increase sodium reabsorption and potassium secretion. There is also calcium reabsorption, where the calcium is reabsorbed through the calcium channels. This time, the parathyroid hormone influences this, thereby leading to elevated calcium levels in the blood. And then another clinical principle is that the distal convoluted tubuli is the site of action for thiazide diuretics. And one classic example is dihydrochlorothiazide. By the way, I forgot to mention that diuretics are medications that help to lower blood pressure with a side effect of urinating a lot more. So patients who take these medications will frequently report that they pee a lot, or at least they pee more than usual. Alright, so collecting duct will follow the distal convoluted tubuli, and remember that the collecting duct is over here. Once again, water is reabsorbed, as the collecting duct is permeable to water in the presence of antidiuretic hormone. This allows for the final concentration of urine. based regulation occurring in the collecting duct, specifically through the secretion of your H plus proton and the reabsorption of bicarbonate. Once again, this is important for maintaining pH balance, and the H plus proton will be secreted through the H plus ATPase channel and the H plus K plus ATPase coupled channel. Okay, now that we understand, or at least have more information of what goes on in each part of the nephron, let's talk about the regulation of renal function. So the kidneys have multiple mechanisms for regulating fluid and electrolyte balance. And one classic example is the renin-angiotensin-aldosterone system, or RAS. So renin is secreted by the juxtaglomerular cells in response to low blood pressure, low sodium, or sympathetic stimulation. So remember that in response to low blood pressure, low sodium, or sympathetic stimulation, the renin will be secreted by the juxtaglomerular cells. After being secreted by the juxtaglomerular cells, the renin will be responsible for converting angiotensinogen to angiotensin 1. Afterwards, angiotensin 1 will subsequently be converted to angiotensin 2, and this time it is the angiotensin converting enzyme that is responsible for converting your angiotensin 1 to angiotensin 2. The angiotensin converting enzyme, also known as ACE, comes from the lungs. And finally, when we have angiotensin 2, this will stimulate aldosterone release from the adrenal cortex. And remember that we said that aldosterone is responsible for increasing sodium reabsorption in the distal convoluted tubuli and collecting duct. Angiotensin 2 will also constrict the efferent arterioles, which will help to decrease the glomerular filtration rate. It will also stimulate antidiuretic hormone release from the posterior pituitary, thereby increasing water reabsorption. And finally, it's going to induce thirst while increasing sympathetic tone. So these are some remarkable features of the hormone angiotensin 2, or the enzyme, the product angiotensin 2. Okay, what else is there besides the RAS system? We have this peptide called the atrial natriuretic peptide, or ANP. So the atrial natriuretic peptide is released from the heart in response to atrial stretch. And the atrial stretch is due to the increased blood volume in the heart. And once the atrial natriuretic peptide is released, it promotes natriuresis, which is another way to say the excretion of sodium. When sodium is excreted from the blood system, this will result in decreased blood volume and pressure because water or fluid tends to follow wherever sodium goes, once again due to osmolarity. So if there's more water and fluid escaping the blood, that's going to decrease the blood volume, and as a result, decrease in blood pressure. Makes sense intuitively. Interestingly, the atrial natriuretic peptide will inhibit the renin release and ultimately reduce sodium reabsorption in the collecting duct. So, in a nutshell, the atrial natriuretic peptide is released because the body basically says the blood pressure is too much. On the other hand, the renin-angiotensin-aldosterone system is triggered whenever the body thinks that blood pressure is too low. So always remember that these work the luminal membrane, also known as the membrane adjacent to where the urine is being concentrated. And when you have water being reabsorbed, the urine becomes more concentrated. Alright, now that we've gone over the different structures of the nephron and how they're regulated, let's now go over the electrolyte and acid-base balance associated with the kidneys. The first and very important electrolyte is sodium. The kidneys regulate sodium balance through the reabsorption mechanisms in the proximal convoluted tubuli, the loop of Henle, the distal convoluted tubuli, and the collecting duct. This is primarily in response to the renin-angiotensin aldosterone system and then your hormone aldosterone. Potassium is another important electrolyte. The kidneys excrete potassium primarily in the distal tubuli and the collecting duct, regulated by aldosterone once again. Then we have calcium, where calcium balance is regulated by the parathyroid hormone and vitamin D, with reabsorption occurring in the proximal convoluted tubuli, the loop of Henle, and in the distal convoluted tubuli. And finally, regarding acid-base balance, the kidneys excrete hydrogen ions, or H+ ions, and reabsorb bicarbonate in order to maintain pH within a normal range, which is around 7.35 to 7.45. Why is this important? Because the blood needs to be buffered, and so that its function will be maintained. Alright, let's talk about another important principle, which is renal clearance and transport. So renal clearance, or kidney clearance, refers to the volume of plasma where a substance is completely removed by the kidneys per unit of time. The formula you need to know is C equals U times V divided by P. C stands for clearance, U is the urine concentration of the substance, and then V stands for the urine flow rate, and lastly, P stands for the plasma concentration. Once again, you have to multiply the urine concentration of the substance by the urine flow rate, divide the product by the plasma concentration. And when you do this mathematical operation, you can calculate the clearance. And ultimately, calculation of clearance can help you to determine whether your kidney is functioning properly and is ultimately healthy. So, now when you are running out of time and you need to estimate the glomerular filtration rate, you use this substance called inulin to accurately measure clearance. This is because inulin is freely filtered and is never reabsorbed nor secreted, so that can be an accurate marker for measuring the glomerular filtration rate. Finally, the para-aminohippuric acid, or PAH, clearance is used to estimate the renal plasma flow, or the RPF. So inulin for estimating GFR, and para-aminohippuric acid for estimating renal plasma flow. Alright, lastly, we have some renal pathophysiology like, and subscribe. And I'll see you next time. Peace out.